All right. Good afternoon, everyone. Thank you so much for coming to our session on human neuroscience in the wild. My name is Corey Inman. I'm an assistant professor at the University of Utah, and it is a distinct honor to be here today. Thank you to the organizers of the Brain Initiative meeting for inviting us to present this symposium. We're very excited to share our work with you. Today, you'll hear from myself, Nanthia Suthana from UCLA, Nick Turk-Brown from Yale, and Nicole Provenza from Baylor. And we'll be going in that order. So first, I'd like to talk and just do a bit of an introduction to why do we want to think about doing human neuroscience in the wild? Where are we in the field? What technology and tools have become available to us? And what is the sort of psychological or sort of neuroscience motivation for the work that we want to be doing? So first, I'd like to, a big thesis in my lab's work, and I think a lot of people's work, is that cognition and behavior really occur in the dynamic real world, and that oftentimes, due to the limitations of traditional neuroimaging methods, we have been constrained from being able to do studies in the real world with people moving freely and dealing with all of the sort of complexity that comes with our natural behaviors in the real world environment. So functional MRI, MEG, scalp EEG, even intracranial EEG in patients often require immobility, and in the case of intracranial EEG, they are literally tethered to the wall. So they really can't go very far. They can move a bit more than you can in, say, fMRI, where you can only move 2 millimeters in any direction, and then the data is no good. So these are often require immobility, and due to movement artifacts, they are either recording activity from superficial brain areas or at slower temporal resolutions. And thus, because of this, we've had to use fairly unnatural behavioral tasks. They're often low in ecological validity. Now, of course, everything isn't about ecological validity, but there's still a lot that we need to understand about what is important about how the brain operates in the real world relative to how it operates in our controlled laboratory studies. So there are lots of new opportunities for doing cognitive neuroscience in the wild, or just neuroscience in the wild more generally in humans. There's mobile eye tracking, small, high-quality cameras that have gone through multiple generations and iterations and developed, to be very robust in the real world. There's mobile movement tracking that's been used in Marvel movies and all kinds of other movies to make characters and things like that. So very precise models of the human body and how it moves through space. There's GPS tracking on all of our phones, and lots of other capabilities that our phones have in terms of being able to record various types of sensors about our experience. There's augmented reality. We can present things in people's actual real world experience. In fact, many of you in this audience might have played Pokemon Go a few years ago, or maybe you're still playing. That was an augmented reality experience that allows you to put things into your own world. So you could imagine building tasks where you have to remember where you caught said Pokemon or the shiny Pokemon or whatever it may be. And really, another huge innovation is the fact that we now have patients that have devices implanted in their brain, whether they have epilepsy or OCD or other FDA-indicated devices or approved devices. So the two that we'll be talking and hearing about today are the NeuroPace responsive neurostimulation system that was approved by the FDA in 2013. And there's currently over 2,000 people, probably more on the order of 2,500 people at this point, implanted with this device. It's implanted in the skull. It's got 2 leads that have 4 electrodes apiece that are bipolar referenced. So they're referenced to one another. So that gives you essentially 4 sensors of local field potential activity coming off of the human brain. And these are often implanted in patients that have intractable epilepsy, for which they've determined that resection may not be the best option. Say both hippocampi have seizures starting at around the same time, so they have electrodes in those areas like the hippocampus. So we have lots of patients that have good coverage in memory-related areas. And then the Medtronic Summit RC plus S and the PC plus S. systems that you'll hear about from Nicole Provenza's talk that has some very good updated sort of capabilities of doing wireless transfer of data. And you'll see some of the hoops we've jumped through to make this sort of recording work in the RNS patients. And in RNS and in the RC plus S system, these electrodes are implanted in very precise regions that are related to the disorder of interest, whether it be epilepsy or OCD, Parkinson's, and so forth. So Nanthia Suthana has done some of the pioneering work to make these sorts of ideas possible, and I was very fortunate to work in her lab and have now started my own lab to do this type of work. And in 2017, she and Zara Argajan demonstrated, that when a human is walking faster or slower in the world, that their hippocampal theta changes. And then we've recently published a paper showing how this system was built in Uroš Topalović's work. And so you can get these nice local field potentials, theta activity from the human brain as people walk around. All right, so why do we want to do this type of work? Well, in my opinion, I think that this is one of the ultimate goals of cognitive neuroscience and maybe neuroscience more generally, that we're ultimately trying to explain how the human brain works in the real world. That many of the things that we want to understand about cognition are fundamentally human. The amount of time that we can think into the future or into our past, the ability to subjectively describe what we experience and feel, the current limitations that I mentioned earlier. And for me, a big reason for this is if we're ever going to realize the promise of neuromodulation, we need to start trying to understand how the brain works in the real world. And so we can work from these highly controlled laboratory experiments out towards the natural environment where we have higher ecological validity but less control. We can do that through VR and augmented reality and sort of draw this bridge, but we don't necessarily have to do this incrementally, one after the other. We can actually have studies of how the brain responds in the natural environment now and have that inform the studies that we do and create this sort of positive feedback loop to try to figure out how the brain is operating in the real world and how it informs our laboratory experiments. And I would like to also say that, with new tools, we have to incentivize observation, taking that step back in the scientific method, back from hypothesis testing to observation, just to see when we have these new tools, what do we see when people are doing real world behaviors? And these new tools offer a new observational perspective on theoretical questions. But currently this hypothesis testing is the primarily incentivized in funding and publication. So what I'd like to call for is us to be really thinking about doing a truly exploratory work that allows us to fully take advantage of these new observational opportunities. And that it's incentivizing these observational or exploratory studies of real world behaviors in complex tasks will allow for better informed hypothesis testing. I think the BRAIN Initiative has been a pioneer in making that type of work happen, and I'm very excited that we get to present this work here today. All right, so now I'll move on to my specific talk. That was more sort of general introduction for everybody's talks to minimize having to repeat ourselves. So today I'll be telling you about some of our work demonstrating the environmental context shifts and modulates human temporal lobe activity during real-world navigation. So we know that memories form during continuous, freely moving behaviors and that they're made at the spatial and temporal scales of our daily lives. There's been excellent work in the fMRI realm where we've had people wearing cell phones and you can document where they were and when they were there. And you can have them retrieve those memories in the scanner. However, we have not had any studies of being able to have people make memories in the real world and be able to record from their brain as we do that. And so, there are lots of questions about that there's lots of different angles to get at this question from. We started with this perspective that we don't even know what the event structure is at this point of our continuous experiences. There's been some great theoretical work that has demonstrated that, and behavioral work in the form of event segmentation theory. So what this model suggests is that spatial shifts or goal shifts or perceptual shifts lead to segment segmentations of our experience into event boundaries and into episodes, right? These are the episodes that we may be able to reconstruct later on. And that behaviorally, in terms of memory, we know that across these event boundaries, that order memory is impaired, that you have increased temporal distance estimates and overestimated duration of the entire experience. And you also have enhanced item to context binding and item recognition around these event boundaries. This has also been demonstrated when you have people watch movies of people doing everyday actions or having them watch Forrest Gump or a Alfred Hitchcock film. And what you can find is that there's agreement on when people feel like a meaningful unit ended and another began across individuals, suggesting that there's something that we're picking up on as individuals that demarks the end of one event and the beginning of another. And when you look at that sort of activity in an MRI scanner, with people you have one group of people that are just watching the movies in the MRI scanner, and then another group of people that are that have made these sort of annotations of when they feel like one meaningful event ended and not. When you have high agreement or high salience in this case, you get increases in hippocampal activity. And so we wanted to take this idea and expand it out to what our naturalistic experiences are, right? In a movie, it's going to be a directed, edited movie with scene shifts and things like that, but that's not how we experience the world. As you went through the poster session this morning, you were going from poster to poster, and you had maybe a little few breaks in between where you kind of checked out for a second and maybe were thinking about something later on or an email. But really, you were continuously in your body experiencing your first-person perspective. And so what we want to think about here is sort of spatiotemporal or neural context drift. So the faster that happens, that's where we would expect these event boundaries to occur. So these can be defined based off of a group of observers or having people make turns along an experience or going from one room to the next, or say going from indoors to outdoors. And what we expect to see is that we would see changes in hippocampal theta. This comes from some of the work that we've done in Northeast Silana's lab with Matthias Stangl that showed that as people approach the walls of a room, that you see increases in theta activity. But we wanted to ask, what if you walked through the wall into the next thing that you can't predict, right? So another model of this is that when you don't have the ability to predict what's going to happen next, that you get these shifts or remapping of hippocampal activity. And of course, it's not just the hippocampus, it's an entire posterior medial network that has been demonstrated across many studies, but we think that the hippocampus is a key determinant of when these boundaries are detected. And so what we asked people to do is first we developed this thing we call the photon pack. This is one of our patients wearing it. It's got a GoPro and skin conductance monitors, heart rate, eye tracking. This is the wand that allows us to record from the NeuroPace device. We have a Raspberry Pi here that helps us synchronize all of these devices together. And so what we had people do is just do the most naturalistic possible task we possibly can have them do, which is we teach them a maze around the UCLA medical campus, which is a maze in and of itself, where they have as many turns and indoor-outdoor transitions as possible. We had them walk this route where I guide them once. We had them then walk the route with them navigating, and we allowed them to get lost. We're interested in those moments in time where they get lost and they realize, oh no, I'm going the wrong way, or when we have to correct them. So we had them do that 3 or 4 times. We also do some control walks because we know that things like velocity of movement modulates theta. So we had them stop at particular points in the task and keep moving during particular points in the task. And the last walk that they did, the 7th or 8th walk, they started at the end and navigated themselves back to the beginning in order to sort of reset their mental map and see if they actually had a good understanding of the environment. We can also do memory tests. So we had landmarks that we documented along the way, and we can test their recognition memory for that later on and look at the eye tracking data from when they first looked at that item to do subsequent memory analyses as you would do in a typical fMRI study. And then we've also had 400 students go through and watch these videos to do event segmentation, as I described earlier. So this is what the data looks like from the chest-mounted GoPro camera and the head-mounted eye tracking. You'll see the eye tracking sort of fixation here. You'll see this is the mark that's made in our LFP. It took about 2 years to get all of this synchronization stuff up and running and together, and a whole pandemic essentially, to get it all really working. But with the right engineers and enough gumption, you can get a lot of things figured out. And so we've had 5 participants do this task so far, with 38 total walks, and I like to measure my data in miles, so we have 28.5 miles of human hippocampal real-world recordings at this point. So this is our entourage that's walking with the 360 camera, and the mobile lab behind us that's making sure all the recordings are continuing to work. Then we can move directly in the same day to a laboratory-based experiment, on a computer, just as we would in any of our other patients. So now for some data. So you'll see this is from a lateral temporal cortex electrode. You'll see some sort of theta and alpha activity throughout the 20 minutes of each of one of our walks. Gray indicates when they're indoors, green indicates when they're outdoors, and the black lines indicates when they're going through some sort of spatial transition, say a doorway, or going up a few stairs. And as you can see just by eye, you can see that there's a difference between when they're indoors here and when they're outdoors over here. And so we wanted to quantify that just to look at the contextual change in human hippocampal theta as between when you're indoors and outdoors. And I'd like to suggest that this is something that's even difficult to do in animal studies, because it's rare that you're going to have an animal running around outside and then coming back inside and running around inside. So what we see in a couple individual, you can see nice peaks in this sort of 5 to 8 hertz theta range in both temporal cortex and anterior hippocampus. This is just a couple examples. You also see a peak shift where for both, for outdoor, when you're outdoors, you tend to see higher theta power relative to being indoors. And we see that across 16 of our 20 electrodes. And there's 4 electrodes that that have the opposite effect. These 4 electrodes are all in the anterior hippocampus, are all in the hippocampus. And these are mostly in the hippocampus, and there's a few in the temporal cortex. So this is an interesting insight that suggests that the environment that we are in actually modulates our brain activity. We can also do things like control for velocity and see if this indoor-outdoor distinction is still a significant predictor, which it is. And when you control for the indoor-outdoor distinction, velocity is also a significant predictor, suggesting that these are potentially independent influences on this 5 to 8 hertz theta activity. And we have, of course, a lot of other activity that we want to correlate with this to see, to put into our model to see, does it explain any of the variance in the theta power? So things like eye saccadic movements, their people's ability or their gait rhythm, and so forth. All right, and so my final data slide, we can also do these event-related analyses where we align all of the data, because we've synchronized all of our data down to the sampling rate of every one of our recording devices and all of our sensors. And so we can align the data directly with when they went through particular doorways, say from indoors to outdoors, or from outdoors to indoors. And as you can see, when you do that, you see a very nice, clear change right at the spatial boundary in higher theta and lower theta. And then later on, after about 5 seconds later, you see this decrease in theta power and an increase in gamma power. This beta power does not survive a leave one out analysis. So really pay attention to this bit here. And then we see a different pattern here. And so there's still a lot of things to try to assess with this, but this is all very enticing. This is at the group level across our patients. So there's still a lot of digging to do here, but we're excited to have done this sort of exploratory analysis in a truly naturalistic experiment, so that we can have a good endpoint from which to take the step back to do hypothesis testing, say in VR and so forth, to really try to nail down what factors and variables contribute to these particular findings. So I'll leave this for people to take a picture for a second, but I'm not going to speak through each one of these for sake of time. And I would, of course, like to, I can go back for a second. But of course, I'm just a representative of an incredible group of people that have, one, been the people doing this work in terms of my lab at the University of Utah and my collaborators. And then, of course, the people that mentored me along the way and allowed me to explore this fascinating space. And I can't thank my mentors enough. So thank you so much. So we're going to hold questions till the end, and we're going to keep moving on with the talks, and we'll have a great discussion at the end.
All right, that's there. Okay, I'm going to put this down. I'm not using it. Thank you, Corey, for organizing this, and Brain Initiative for allowing us to come together and talk about these very exciting, I think, topics. So today I'm going to show you some unpublished data. So it's brand new. Welcome your feedback afterwards if you have any time. So I took one of the videos that Corey showed that talking about these dynamic experiences we have in the real world and the fact that we can replay these in our mind, right? We can retrieve them as memories. We can also imagine them. And, you know, this process is also in humans something we can do on demand. We can ask somebody to do this and then measure, you know, the brain activity and try to understand. So there's a really great opportunity here. And we've also summarized our perspective on this in a recent paper across not just memory, but social behaviors, emotion, affect, movement related. If you're interested, take a look further there. So as Corey already reviewed, I won't belabor this, but here's just an example of one of our recordings where you can see the raw data, eye tracking and gaze and position. And I'll show you some data today about imagination as well. So theta oscillations, as Corey nicely introduced, in humans, it comes in bouts, as opposed to rodents where it's continuous as they're running and relates to speed. And in humans, when we first saw this, it's not quite the same. And we've been trying to since then understand why, and what are the behavioral variables that modulate its presence, which Corey alluded to Zara's work, who showed that speed can modulate these bouts, and so faster speeds showing more prevalent bouts compared to the slower speeds, along with the 16 hertz harmonic, which we see in multiple data sets. So Sabrina, oh, is this old slide? It's a slightly old deck, but that's okay. Sabrina Mayos, who's actually here in the audience, and if you have questions, can ask her at the end, designed a study where we used VR. And VR is very useful because you can immediately change the context visually and have a person walk through. I'll play a video of the task that she used in these participants where they had to learn the position of those cylinders, these halos, which are each individually colored. And as you change context, they have unique positions and unique colors. During the retrieval period, you can then ask these participants to try to go and remember where they saw these halos, these cylinders, and so they can walk through and press a button at the position where they remember their locations. You can nicely test memory in this context, and using VR very quickly shift between contexts. Here shows a stone room versus another one you can see in the far right that's a wooden room. Okay, so in this task, participants are able to learn the positions. You can see their memory gets better. Do I have a laser pointer up here? Their memory gets better on the far right from the last block compared to the first block. And if you look at it in a different way and look at their accuracy, so their position, where they recall it compared to the actual location, you see that also improves. So altogether over the task, each color showing a different participant, they're getting better over time. And so what happens in the brain? So when you look at the medial temporal or hippocampal entorhinal theta activity, you see that it's really just increasing during the trials where they're correct, where they're successfully remembering these locations, not when they're incorrectly remembering these locations. And this is occurring around 6 to 8 hertz. And this is not occurring also during some control conditions where they see the halo, so the visible feedback condition, or when they're doing another non-mnemonic task with searching for arrows. So it's specific to successful memory retrieval, and this is showing examples of where the hotspots or the increases in theta activity are happening, with the circles signifying the recall points where they're pressing the button for the different colored halos, and you see them nicely matched across the positions in the successful correct condition, but not in the incorrect or visible in arrow conditions. Now, when you look at it in a slightly different way, you can see the temporal time course of this response, which is happening before button press. So it's increasing right before they essentially make that button press signifying recall, and again, only in the green condition here, which is signifying correct trials. Sabrina also put all of these variables in a linear mixed model, which you just heard that Corey did in his data, and found that performance, or actually whether they're correct or not, as well as something not shown here, trial error, how well they're doing on the task, were the significant predictors of theta activity, as opposed to other variables like speed, head direction, angular velocity, and several others, which I won't have time to show. Another interesting result that she found was that as they're moving along to retrieve a certain halo, like this yellow halo shown here, they might incidentally cross other positions that they learned about in previous trials that had other halos, the red one and the green one. So these non-target incidental halos, as they're crossing through, you see theta increases at those moments as well. Very similar to what you're seeing in rodent recordings that referred to object memory trace responses in single units. And so this is just showing here the increase when you're close to these incidental non-target halos in theta power compared to when you're far away. And this is showing example trials that this is context specific. So if you switch context from wooden to stone rooms, that map remaps and is specific to the context, which is another powerful, useful way to use VR. And then I wanted to present another study. Sorry, some of my pictures are not here, but Martin Sieber in the lab used a navigation task where they had to walk different trajectories that you can see here if the video starts working, where they're either going to the right or the left. And what's interesting about this task is they have to make very specific turns that have specific angles 4 times in each direction. And they do this repeatedly. In the beginning, they are not so good at it. And you can see on the left, their performance is wavering. They get feedback after each trial, showing them the correct trajectory compared to their trajectory. So they get better. As you see on the right, they're really following this trajectory very nicely, and across 5 participants on the top, they're learning these trajectories quite well. So what's interesting about this study is in the middle, at the end, the person is standing on a treadmill and walking. And we asked them in the beginning, just walk, don't have to do anything. But in the latter half of the trials, we asked them to imagine the past trajectory versus the upcoming trajectory, and we randomize this, left or right turns. So this is a unique period where you can literally instruct human participants to just replay or imagine the experience, at this moment. And so what's happening? In the navigation condition, you see across all the participants that theta is modulated by the turns. And these are very critical retrieval points because they have to remember the exact angle. And this is just showing the data from each one, and you can see it just by eye that the theta activity is increasing right before basically each of these turns. Even in one participant, P2, who added an additional turn because for whatever reason they wanted to do this, and you see again that hotspot in that additional turn that's not there for the other participants. If you look across all the data and align it to the turns, you see that this theta activity is increasing right before the turns, even though speed is going down as they're turning. And we also have eye movement data, and there's some very interesting eye movement relationships to this that I don't have time to present, but happy to discuss. So during Imagine navigation, we see that this effect is essentially recapitulated. And so if you map out the data according to basically the entire maze trace, right or left turns, you see there's increases around the turn points. And if you look across trials at the temporal consistency, you see it's significantly above chance, the control condition being there's just no temporal consistency across trials, versus in imagination, there's a significant effect, and as, of course, in actual navigation, it's even higher. And the relationship between imagine walks and actual walks is significant, such that the temporal consistency across trials, if it's stronger in a channel during navigation, it's also stronger during imagination, and the channels that show the strongest effect are in the anterior hippocampus. So to summarize all of these findings, we see that this theta effect is modulated by successful memory retrieval. It also is modulated by these object trace type of effects, which may reflect past relevant experience of learning these objects in these positions. We previously published that it can be modulated by distance to walls, as Corey mentioned, and also by the position of another person near, relative to walls. And then lastly, I just showed you that it can be recapitulated or internally generated in the absence of any bodily or visual cues, right? So they're walking on this treadmill, but external sensory cues are not present. And so this suggests that this theta activity is very much modulated by the task, can be internally generated, and sort of basically depends on the behavioral demands. Now what we're doing is we're recording single neurons. Uroš Topolovich in the lab has led an effort by many, many engineers in collaboration with Dan Markovich at UCLA to develop a handheld system called the Neurostack, which you see here, that can be worn in a backpack on the body with patients who are in the epilepsy monitoring unit who have microwire electrodes implanted that can record single unit activity. We had 6 participants walk in the hospital room while this device was connected to those electrodes and recorded single unit activity, which you can see here. And now we're doing this with participants walking longer routes in the hallway of the hospital. So we're very excited to start to combine some of these methods with single unit recordings and moving humans. So with that, I'd just like to thank all the folks that are doing this work, especially these first 4 here that I presented data for. And Sabrina, like I said, is in the audience and will be here afterwards to chat. And thank you all for the collaborators and funding and many, many people who have contributed to this work. And with that, I'll end here. Thank you.
Okay, thank you to Corey and to the Brain Initiative for having me here, and thank you to Nantia for saving me a lot of time with her beautiful presentation. I'm going to talk today about the work of a graduate student, Kat Graves. She's a D-SPAN F99K00 recipient, about to head off on her postdoc, and also our clinical collaborator who's in the audience up front here, Dr. Imran Qureshi. My lab studies human learning and memory, in particular, thinking about the ways in which these aspects of cognition interact with other components such as perception, attention, decision-making, and others. And the reason why these different aspects of the mind interact is because in the real world, we're using all of them simultaneously. So it's a bit naive on my part then to study this in the context of fMRI, which really limits behavior, or even the work we do in the hospital with intracranial EEG. The kinds of RNS technologies that Corey introduced provide a really beautiful platform for studying cognition in the wild. What I'm going to tell you about today is some of our early studies using RNS to essentially validate this method for uncovering some of the properties of the hippocampus that have been observed with other methods, both in humans and in animals. In particular, I hope to show you how the hippocampus codes for location, direction, and speed during real-world human navigation, to identify some of the frequency bands of activity that drive these representations, and to do a little preliminary work to look at how stable they are over time by testing patients longitudinally. I'm going to present data from 12 RNS patients, all of whom had at least one electrode in the hippocampus and most bilateral. And they did a very, very simple experiment, ironically, for studying the real world, where they came in and they sat in a chair and we played them one of 2 songs. And then we had them walk back and forth on a linear track. Critically, when they heard one song, the green song in this case, they were to walk back and forth. And when it switched to the blue song, they were told to stand still in place. And they alternated back and forth between walking and standing for a minute each, a total of 20 minutes. Afterwards, they sat back down in the chair and we played the songs again. And what I'm going to focus on today is in the middle part of this task, where they were walking back and forth and standing, but I'll also show at the end some preliminary data from the pre-post comparison of what kinds of hippocampal signals are elicited by songs before and after they're paired with navigation. I'm not going to spend a huge amount of time on the setup because you've seen this already, other than to thank Nanthi and her group, who were very, very generous in helping us get set up a few years ago. This is our environment. It's a multimedia studio. Those little white dots are footsteps, so that's where the patient would walk back and forth on a 32-foot linear track. And what you'll see on the wall, there are some projected images to provide at least rudimentary landmarks as they navigated back and forth. Here's a video depicting Kat doing this task wearing the setup. So you can hear the walking song played in the background. That's not flash photography. I think it's a hiccup in the video. Okay, I'm going to show you essentially four pieces of data from this navigation phase. The first is the crudest, which is attempting to decode whether the patient was walking or standing. And so here we map their actual movement shown in the trajectory at the top onto a linear track. You can see the time course presented here, and so those plateaus are points where they're standing. We label the spectral decomposition of LFP from the hippocampus based on standing versus walking, and use leave one patient out cross-validation to try to decode using a support vector classifier which state they're in. Here's an example of the average power at the frequency range that we're covering over time. In particular, we're looking at decoding the onset of movement versus standing. And indeed, we can decode this quite reliably across patients, so training on 11 patients, testing on the held-out patient. Importantly, when we do a nested cross-validation procedure within the training set, we find that this decoding is driven entirely by signals in the theta band, consistent with some of what you saw earlier. Okay, let's get a little more granular. And so in this case, we're going to label time points not based on walking versus standing, but during walking or standing, which direction the patient was heading. And again, we trained a classifier to decode heading direction and found that during walking, we could reliably decode which way they were going. However, when they're standing and facing in those same directions, we could not decode which way they're moving. So this likely reflects some kind of integration of landmarks with self-motion that drives classification. Again, signals in the theta band drove this performance. I'm showing a bunch of other data here, but for the sake of time, I'll just point out over here. We had 4 patients who we invited back between 4 days and 3 months later, and we had them do the same task. What's shown in the bottom right corner is that we used the models that we trained in the first session to try to decode heading direction in the second session, and could reliably do so in those 4 patients, suggesting that these sort of spectral features coding for heading direction are stable over time. Place coding was another obvious thing to look for here. Instead of doing classification, now we're going to use a regularized linear regression model to predict the x-coordinate on the linear track. And what I'm showing you again here is our ability to do a cross-patient prediction of location on the track based on spectral features. Again, we reliably were able to do this, and the signals that allowed us to best predict location were in the theta band. Interestingly, in this case, and consistent with animal work, we were not able to decode across session the position of the patient on the track. That is, the models trained in their first session did not generalize to the second, suggesting some remapping, even though it was the same environment, perhaps temporal context or other aspects of the day. The final sort of navigation signal I'll show you is speed information. So again, we extracted instantaneous speed using a positional tracker and used a regularized linear regression model to predict their instantaneous speed. And what I'm showing you here is our ability to predict speed across patients based on spectral features. And again, this was highly reliable. In this case, the best frequency bands for predicting speed was the whole broadband signal as opposed to limited to the theta band. And just like with heading direction, now we could actually decode speed across sessions within channel. Okay, so just to show you a bit of what this looks like, this is just a snippet of behavior. What's plotted on the left is walking. The bullseye reflects our predicted place based on hippocampal signals. What's plotted on the top is the heading direction, northeast versus southwest, and in the bottom right, instantaneous speed. So our ability to track these things is what drove the classification and regression prediction performance that I showed before, but this is a visualization that we can get at an individual patient's individual channels. Okay, final thing I'll show you is, again, not just the middle part here where we were able to decode walking versus standing, but we wanted to use this task as a platform for beginning to investigate some of what Nanthia was discussing, which is the remarkable ability of humans to mentally time travel, to revisit moments in the past in our mind's eye. And so here we're using the songs as a way to perhaps evoke navigation-related signals. Before they did any track navigation, we could not decode the identity of the song based on hippocampal activity. But after navigation, after these songs have been associated with movement, even though they're sitting in a chair, we could now decode reliably that they were moving when they heard the movement-related song. And in fact, if you look just within the theta band, this effect is much stronger. So I'm going to stop there and just summarize, say we found evidence for these spatial codes in the human hippocampus during real-world navigation. I didn't have time to go into it, but these codes are multiplexed within channels and even within frequency bands within patients. There seems to be an emphasis on the theta band, at least for direction and place coding, as has been mentioned before. There's evidence for stability of direction and speed coding, but not location. And finally, some early evidence using auditory cues to reinstate perhaps memories of navigation. And now we're engaged in more complex tasks to use these spatial signals to try to understand the nature of learning and memory in humans. For example, how memories are transformed over time from memory for discrete episodes to more generalized statistical knowledge. And Kat has done some really beautiful work in both human behavior and human intracranial recordings, looking at these kinds of memory transformations, which we can now explore in the real world. So I'll stop there and thank my lab and thank all of you.
Hi, everyone. I'm Nicole Provenza. I'm a postdoc in Samir Shah's lab at Baylor College of Medicine in Houston, Texas. Thank you so much, Corey, for organizing this. I'm so excited to be here. I'm going to be talking about my work on identifying neural biomarkers of OCD symptom intensity in response to deep brain stimulation for OCD. And before I get into it, I just want to thank our awesome team at Baylor College of Medicine that makes this work possible. These are wonderful team members, and the work I'm presenting today is part of a large collaborative NIH-UH3 brain initiative effort led by Wayne Goodman. And here are awesome collaborators, including Jeff Herron at the University of Washington, Jeffrey Cohn at the University of Pittsburgh, and David Borton at my alma mater, Brown University. So the overarching goal of my work is to improve the efficacy of deep brain stimulation for treatment-resistant OCD. So DBS of the ventral capsule ventral striatum, or VCVS, provides clinical benefit in about every 2 out of 3 treatment-resistant patients. So how can we make these outcomes more consistent? One improvement area revolves around DBS programming. So the current programming paradigm for OCD is open loop, requiring visits to the clinician's office after DBS is turned on for the first time to optimize stimulation parameters. So the patient comes into the clinic, stimulation is turned on or adjusted, and they might say, I think I feel better. But then they go home and they might experience side effects of DBS of this region or no improvement in OCD symptoms. They go back into the clinic weeks to months later and they don't feel better anymore. So we're proposing that automatic detection of mental states related to symptoms and side effects might enable data-driven intervention. And ultimately, our goal is to develop an adaptive system in which neural biomarkers of relevant symptoms or behavioral states are used to control stimulation on the device in an adaptive ADBS way. So a critical component of this work is figuring out what's going on at home or in the wild, where the fluctuations in symptoms and side effects are actually taking place. So the goal of the adaptive system would be to find a balance between treating OCD symptoms without inducing side effects. So if stimulation amplitude is set too low, you might still have OCD symptoms, but if it's set too high, you might start to see side effects, namely hypomania, which is marked by decreased need for sleep, increased risk-taking and impulsive behaviors, feeling of grandiosity. So the optimal stimulation amplitude setting is kind of like a sweet spot where the desired state, which involves increased mood, increased approach behaviors, and increased engagement, sits somewhere in between these two extremes and might fluctuate over time. So our goal is to identify neural biomarkers of these two states. So in this talk, I'll present first on our work on identifying neural biomarkers of OCD symptom severity during provocations at home. And next, I'll talk about some unpublished data on identifying neural biomarkers of increased mood and engagement induced by acute stimulation changes in the clinic. So in order to identify biomarkers to enable an adaptive system, we need to record neural data at home where these fluctuations in symptoms and side effects are actually happening. So to that end, we're streaming intracranial local field potential data from the investigational Medtronic Summit RC+S device in both the clinic and at home, time synced with OCD relevant behaviors. So participants control their own recordings at home by using a patient-facing application that was developed at Brown while I was there, and they log OCD symptom intensity from 0 to 10 on their phone using this application developed by Rune Labs using this 0 to 10 slider, where 0 is no or little symptoms and 10 is extreme symptoms. So one way we try to maximize our recordings at home is by conducting multi-day marathon recordings where the patient's asked to record for a few days with breaks for recharging devices. So this participant did a 3-day marathon recording, which is shown here in this timeline, where there were natural exposures to OCD symptoms on days 1 and 2. So those are circled. And so this recording stood out as a really great opportunity for analysis because we don't often see such wide fluctuations in OCD symptoms during recordings. So we're trying to build a classifier to distinguish between low and high symptoms. So in order to do that, we have to collect a lot of data associated with both states. So here I'm showing LFP data availability in orange over the 80 hours and self-reported intensity of OCD symptoms shown on the y-axis. So to look at the data, we looked at spectral power in canonical frequency bands for the 2-minute segment of data around each symptom intensity rating. And here are the results from that analysis. So there were some interesting correlations between spectral power and OCD symptom intensity across all these frequency bands, where delta band stood out to us. There was a strong negative correlation between delta band power and OCD symptom intensity. And so we were really excited to see this result because this is the first piece of evidence that we found that BCVS activity might be reflecting OCD symptoms. So moving on to the second part of the results. So in addition to investigating biomarkers of OCD-related distress, we need to better understand activity associated with improvement after DBS and hypomania induced by DBS. So in the second set of 5 patients in our study, we added additional sensing electrocorticography electrodes over the orbitofrontal cortex, or OFC, to investigate VCVS and OFC LFP biomarkers simultaneously. And to investigate how changes in DBS amplitude acutely affect neural activity and behavior, we designed this experiment where we changed stimulation amplitude bilaterally from a safe low amplitude, here is 0 milliamps, to a safe high amplitude, here 5 milliamps. And we hold it at the high amplitude for 3 minutes, back down to low for 4, and repeat this again. And so next I'll show you a video of the patient during the time period right around this arrow where stimulation went bilaterally from 0 to 5 milliamps. And by the way, patients are blinded to these stimulation changes. They don't know when stimulation changes occur.
That was kind of cool. I don't know, that was a good little jolt. Whatever that jolt was, I like that. Do more of those jolts.
What was it like?
Oh, just like a little hit of, adrenaline or something. I don't know, something good.
Okay.
But yeah.
So this example is from the very first day of DBS programming for this patient, and something that we noticed throughout this recording in future recordings in this patient and others is that after DBS amplitude increases, patients are more likely to engage in conversation, they start to make more eye contact, make jokes, even when they don't articulate that they've noticed an obvious change, as the patient did here. So what's the neural data telling us? Here I'm showing power spectral density plots with power versus frequency from our 6 recording channels, first VCVS, medial OFC, and then lateral OFC. And this is just DBS off data. And I want to draw your attention to this interesting 30 hertz blip that we see in left and right lateral OFC. When we turn stimulation on, we see this focal 30 hertz power increase in lateral OFC. And we notice that this effect seems to persist into the DBS off period, especially the third off period, after the most cumulative stim. And so our next goal was to see if we could link this result with the behavior that is so obviously captured in the video that I showed, but with audio recordings. So just plotting the raw audio from both the participant and the experimenter, you can see that the audio activity increases during DBS on and fades during DBS off. So looking at just the audio from the participant alone, you can see this also is true for speech rate in words per second and word volume. And so tying it together with the neural activity, you can see that there's this 30 hertz signal that seems to activate when DBS turns on and then trails off after DBS goes off again in a way that actually reflects the speech features. So we're really excited about this result as an early biomarker of improvement after DBS for OCD. So in summary, we measured VCVS LFP during natural OCD symptom provocations at home and during acute bilateral changes in stimulation amplitude in the clinic. And we identified low VCVS delta band power as a state that might indicate need for stimulation intervention. And on the flip side, we identified high 30 hertz power in lateral OFC as a preliminary biomarker of a healthy state or side effects. So with this therapy, we have conflicting priorities of reducing symptoms without producing side effects. And these initial data suggest that biomarkers for these two competing states might be different, which would be really ideal, so we can try to figure out how to simultaneously optimize both. So as we're able to do this more successfully, ultimately these devices are going to affect how patients think and feel without their direct involvement. So we really need to think about how these issues of autonomy and self-determination will impact the patients as we develop closed-loop strategies for psychiatric DBS. So I'll wrap up by talking a bit about where I think we're going next as a field. I think we can do a lot more to beef up our behavioral quantification efforts in the wild. So over the past few years, there's been a ton of advancement in wearables, commercial devices, and sensors that you can use to extract features related to things like daily functioning. And we could apply these sensors across various psychiatric and neurological disorders. Then we could use dimensionality reduction techniques to identify clusters of disease-relevant behaviors that might map to brain states. So I'm really optimistic about the push towards doing neuroscience in the wild, and I think it'll help us better identify biomarkers of disease-relevant behaviors and will ultimately help us improve patient outcomes, which motivates us all. So thank you.
Okay, well, we have several minutes for questions. And, you know, I think the first question I'd like to start with, given the excellent emphasis on neuroethics that is essential for all of the work that we're doing, that, you know, I want to sort of ask the panel, you know, kind of what are the sort of ethical questions that you're thinking about in line with your work. You know, I think, you know, at least from my work, you know, thinking about both the patient's privacy and the privacy of other individuals and their agency in sort of making these decisions and understanding, you know, what it means to have neural recordings of memories that they're making as they're moving through the world. And I think there's a lot of work to do on that front. And so we've proposed in one of our recent grants to sort of do a sort of qualitative experiment trying to understand what people's understanding are of what are the brain signals that we're recording. What might they, what might they tell us about the memories that they're storing as they have these real world experiences and their interactions with others. So I'd love to hear what everyone else's perspective is on that. I think you have to hit that.
There we go. So I kind of left off on talking about how we should be thinking about closed loop in terms of, I guess, patient agency. And, if we give the device the control of the stimulation parameters and, maybe the device makes the wrong choice or uses an artifact instead of a real brain signal to control stimulation. And, we end up going in the opposite direction that we wanted to. What are our fail safes? And I'm not sure I have good answers right now. But I think it's something that is important to think about as we develop these closed loop strategies, like having a way for the patient to revert to continuous constant stimulation, for example, could be a backup.
Okay, so now we'd like to open it up to the floor for any questions. I know we might have some online questions. We're going into the break now, but feel free to stick around. We can keep having this conversation and move on when we're done with the questions. Yeah.
Some of the work that some of the speakers presented involved using eye tracking glasses and recording the scene. So this is a bit outside of the realm of people whose brain activity is being recorded, but you're recording other people in the scene who they might be interacting with. When you think about these kinds of research and you think about privacy implications, I think the natural place to focus, justifiably so, is the patient. But have you guys thought in any of your work about the people who are being recorded, their agency, and what is the thinking about that? That's one. I have one more, which is for Nicole specifically, because you're doing closed loop stimulation. You talked a little bit about fail safes, and that's more, I guess, at a mechanistic level, that you don't want to cause any harm. Have you had any occasion to kind of step back and think about, we're basically altering people's mood and state of mind, and how do we think about the ethics of what's okay and what's not okay outside of treating the disorder? Thank you.
Yeah, I'll take the eye tracking one first. So I think this is an excellent question and something that we've thought a lot about. We've had great conversations with our IRB about these things and trying to think about, what is the sort of standard, both legally, what's the standard, and just ethically, what is the standard? And, at this point, in our discussions with our neuroethicist, with our sort of legal scholars on campus, is that public spaces are open, spaces where privacy is essentially, legally at least, not. There is essentially no privacy. We all have our phones. We can take videos and pictures of anything or anyone at any time, essentially, or at least most of the time. And so that's been our sort of standard. Now, that doesn't necessarily mean that needs to be the standard going forward, or that the legal justification of that is best. And I think that that's going to take some sort of understanding and working with community partners, say at like membership events at, say, a garden or a park that's a kind of closed environment, where we can actually do focus groups and try to understand what other people's perspectives are on recording their activities despite not getting their direct consent. But I think that there's a lot of different approaches to doing that, but I do think it will take research to do that. I don't think we can come up with a standard without doing the research first.
And I can answer the question about how we're thinking about developing adaptive DBS in psychiatric disorders, where we're essentially altering someone's mood state. So luckily, we have our colleagues in the movement disorder field who have paved the way for us. I think a lot of ways in psychiatry. There are a lot of differences, but something that we think about a lot is the therapeutic window, where what is the range of DBS parameters that is going to be effective for this patient. And so in movement disorders, they define this therapeutic window based on tremor reduction, for example. In psychiatric disorders, we're still trying to figure out how to define this therapeutic window. Maybe it's an acute effect related to a mood change right after stimulation onset. And we kind of know when the patient gets uncomfortable, for example, if amplitude is set too high. But I think before we move to adaptive DBS, we need to better understand both the short-term and long-term effects that DBS has on someone with OCD or depression, for example.
Excellent. I think there was a question in the back. Yep.
Dr. Provenza, am I saying that correctly? I had a question about the stimulation and the spectrum that you showed. It looked a lot like an artifact, and it only appeared when there was stimulation. So I assume you might be able to explain that. I just want to get around the fact that I guess a biomarker could be just the effect of stimulating the skin or tissue, but do you think that marker is really related to what you see the change in symptomology?
Yeah, it's a great question. I actually thought that this was an artifact when I first looked at the data. But I realized that after DBS turned off, this 30 hertz signal persisted. So it didn't immediately go away. In fact, it took several minutes to go away after the offset of stimulation. And that was DBS off, not even just DBS at 0 milliamps, which can also produce some artifact. It's interesting that our stimulation frequency is 150 hertz and the signal is at 30 hertz. So this could be a 1 to 5 entrainment situation, similar to the 1 to 2 gamma entrainment that some people have seen in SDN and Cortex in Parkinson's patients. But we're still trying to figure that out.
Okay, awesome. Thank you.
All right, I've been informed that we do need to wrap up. So for any other questions, we're more than happy to chat with people individually, and would love to chat with you throughout the rest of the conference. Thank you very much for coming, and have a wonderful conference.